Good day to everyone joining us, and welcome to today's X Talks webinar. Today's talk is entitled Nuclear Receptors and Drug-Drug Interactions, How to Better Understand Potential Toxicity. My name is Sonia Hunt, and it's my pleasure to be your X Talks moderator for today. Today's webinar will run for approximately 60 minutes. This presentation includes a Q&A session with our speaker. This webinar is designed to be interactive, and webinars work best when you're involved. So please feel free to submit questions and comments for our speaker throughout this presentation by using the questions chat box, and we'll try to attend to your questions during the Q&A session. Now, this chat box is located in the control panel, and that's found on the right-hand side of your screen. If you require assistance, please contact me at any time by sending me a message using that chat panel. At this time, all participants are in listen-only mode. Please note that this event will be recorded and made available to you for future streaming on xtalks.com. At this point, I'd like to thank Indigo Biosciences, Inc., who developed the content for this presentation. Indigo Biosciences, Inc. is a leading provider of nuclear receptor and in vitro toxicology solutions that accelerate scientific decision-making. They supplement the world's largest portfolio of nuclear receptor kits and services and in vitro toxicology solutions with greater results, readability, reproducibility, and faster turnaround times. Their solutions plus supportive team and reliable science and platforms aim to reduce the time, cost, and risk associated with the discovery process. Now it's my pleasure to introduce you to your speaker for today's webinar, and he is Dr. Jack van den Heuvel. He is a recognized expert in the field of nuclear receptor biology and toxicology with over 100 peer-reviewed publications. In addition to his role as CSO at Indigo, Jack is a professor at Penn State University, where he is a program coordinator of the undergraduate toxicology program, co-director of the Center of Excellence in Nutrigenomics, and leads an extramural funded research program. Now, before I pass the mic and the controls over to Jack, we have a poll question for our audience members to interact with in real time. That's going to be appearing on your screen right now. And there it is. Now you can vote on this question by clicking on any of the answers. And the question that we have for you is, what is your interest in drug interactions? Very simple one. And you have 4 options below to choose from. Is it screen potential interactions in early drug discovery? Or are you here because of understanding observed interactions? Or is it interest in drug-induced liver injury or general toxicology interest? So please go ahead and cast your votes. As I said, this is done in real time, so I'll be able to close the polls and share the results with everyone and then pass the controls over to Jack. Okay, that looks like it's the majority of our audience. I'm going to leave a few more seconds there for those of you who have not. If you can go ahead and do so now, it'd be greatly appreciated. Okay, fantastic. Thank you everyone for participating in our first poll question. And I'm going to close the polls now, and let's take a look at the results and see who's in our audience. Okay, so our audience are saying 22% are saying that they're here because of screen potential interactions and early drug discovery. Then we have 13% are saying to understand observed interactions, and then 26% are here interested in drug-induced liver injury, and then we have 39% of our audience members are saying general toxicology interests. So thank you everyone for participating in our poll question there. And now I'm going to pass the mic and the controls over to Jack. So Jack, when you're ready, you can begin. But before you start with your presentation, I just want to know what your thoughts are about who we have in the audience today.
Well, thank you, Sonia, and thanks everyone for taking the poll. Well, I must say there wasn't an overwhelming group of people in any one of those 4 questions there. So that tells me that it's a pretty balanced audience, and so I'll take that into account as I'm trying to discuss how we can use some of the tools to look at drug interactions. So without any further ado, let's jump into the content of this talk. So we're going to be talking about nuclear receptors and drug-drug interactions. We're really trying to describe the tools that can best help you understand whether you have a chemical or some potential interaction with a drug or a chemical that you are currently studying. And how we're going to get to the end result of trying to understand drug-drug interactions a little bit more, we're going to start with a basic interactions of what the problem is. I'm going to give a basic overview of nuclear receptors, although I would assume that many of you have a pretty good background in this area. And then we're going to talk about specific types of drug-drug interactions that nuclear receptors participate in. The pharmacodynamic drug-drug interactions, and then the pharmacokinetic drug-drug interactions. And I'll try to talk about some of the key receptors and how we can use some of this information, what approaches we can use, and give some concrete examples. And then at the end, we'll try to wrap everything up, summarize, and then I'll open it up for questions. So let's start with the problem and the nuclear receptor overview. So the problem is increasing in our population as we are aging, and many individuals are taking multiple prescription drugs at one time, and then couple that with different exposures, environmental exposures, and nutritional exposures as well. So this is something that is gaining in an interest within not only the drug discovery area, but also in general healthcare. So whenever individuals are taking multiple medications, we have to be worried about how these are interacting with each other. And so this is the basis of what we're going to be discussing. As far as drug-drug interactions, I will be abbreviating this frequently just as DDI. And we really want to understand how chemicals are interacting with each other so that we can actually predict whether any new chemical that reaches the market, what its potential liabilities are. And we'll be coming back to this particular diagram quite a few times during our talk. And what we're actually talking about in this case is a relatively simple binary question here. Is our drug, and this could be our drug of interest, is its safety and efficacy being affected by some other drug that is being given concurrently? And we'll call this the second drug the perpetrator. And there are really two major ways that this perpetrator drug can affect the activity of our drug of interest. One is pharmacodynamic, and this is your classic pharmacology interaction, where these two drugs are trying to interact with the same molecular target. So we're assuming that whatever drug one is designed to interact with to lead to its beneficial effect, drug two is also interacting with that same molecule. And then again, that's pharmacodynamic or PD drug-drug interaction. Now the pharmacokinetic drug-drug interaction is a little more complicated. In this case, we have our drug one that goes through its normal interaction scheme of, or its normal scheme of activity of, it's absorbed, it's distributed, it's metabolized, and then it's excreted. And all of these factors we know about, and it's used to set the dose and the timing regimen for how we are treating an individual. In this case, our perpetrator drug is somehow affecting one aspect of this absorption, distribution, metabolism of excretion, and then is affecting this balance between safety and efficacy. When we talk about the perpetrators, we've kind of couched this so far, and this is a drug-drug interaction. This is some other purposeful drug that we're giving. But this could just as easily be a drug and environment interaction, where this chemical is a pollutant. It could be a dietary supplement or a dietary component, and this is a drug-nutrient interaction. I will describe at the very end another kind of newly appreciated type of interaction, and that's between a microbial product and some molecular target. And this would be the basis of a drug microbiome interaction. And we know this is a problem just by looking at product inserts for any prescription drug that you take, or talking to your pharmacist or your doctor about concerns about the multiple medications that you may be taking. And this is something that is very difficult to manage sometimes. And these interactions, again, are very well known and are often found on labels of our medications. What you probably don't know from the regular labeling, whether this is a pharmacodynamic or a pharmacokinetic interaction, but they're both present and of importance. So again, with the pharmacodynamic interaction, these are two drugs that are trying to interact with the same molecule, and if they're acting in the same direction, they're both activators of a particular pathway or both inhibitors, then you might have an additive interaction. If you have two chemicals that have opposing effects, then of course this would be an antagonistic interaction, for example, like with levodopa and neuroleptics. Many are pharmacokinetic, and these have to do, again, with somehow affecting the ADME processes of your drug of interest. And this is where you really need to know some of the basics of how your drug is handled in an organism. And these pharmacokinetic inhibitors, this table is showing just one potential site of interaction, and that's with the transporter called ABCB1. And we have substrates, inducers, and inhibitors. And from a pharmacokinetic standpoint, what we're worried about are these chemicals at the bottom, inducers and inhibitors of this particular transporter, affecting the ability to handle the excretion process of the substrates for that transport. And again, we'll go through some more clear-cut examples of this later on in the talk. But obviously, this is a problem that I think we've all been somewhat associated with. Now, one thing that you may not be aware of is this interaction is really talking about two foreign molecules, wherever they're coming from, interacting with specific molecular targets. And the more that we know about what our chemicals are interacting with, the more we can have predictive assays set up, and we can increase the knowledge that we have in making some critical decisions. And really, if you're working in the drug discovery process, understanding potential drug-drug interactions is very important at multiple stages within the drug life cycle, going all the way to the preclinical drug discovery, all the way through the animal testing, through the clinical studies, and then once it gets out into the public, surveillance as well can affect anything as far-ranging as healthcare costs and hospitalization rates. So again, the more we know, the better. And this is true on both sides of the equation. It could be drug one or drug two, your victim or your perpetrator. The more you understand how they're interacting with the cell and how they're affecting cell behavior is going to be of benefit to you. So some of the things that we're going to talk about today are helpful in this drug discovery life cycle. So we can use these approaches for triaging or prioritizing new drugs. We can use this as a means of labeling or putting warnings on any drugs that do make it to the market. And we can also use this for determining the most effective biomarkers that can be used for monitoring both in a clinical study and in post-marketing surveillance. And of course, as the title predicts, we're going to be talking about nuclear receptors and how they are affecting these different types of drug-drug interactions. So why are we focusing on nuclear receptors? Well, first of all, because nuclear receptors are important drug targets. Receptors such as the estrogen receptor, glucocorticoid receptor, PPAR gamma, several others are direct targets that the pharmaceutical industry is screening chemicals for, trying to find new drug candidates and drug leads. Also, the nuclear receptors are very important regulators of drug metabolism, in particular drug metabolism within the liver. So receptors that we'll be describing in detail, things like PXR, CAR, Aryl hydrocarbon receptor, these are very important for driving the kinetic processes of a variety of different xenobiotics. And we're going to try to focus on in vitro approaches to try to better prioritize chemicals, label, and develop biomonitoring tools. Why? Well, cost. We can do some of these in vitro approaches early in the drug discovery process, in the preclinical stage before it goes into animal studies or some of the expensive clinical studies. Most of these assays are medium to high throughput, so we can examine many chemicals concurrently. These are easy assays to run. There are resources available to examine these different receptors that we'll describe, limited expertise and resources that are necessary. And then I'll talk about how we can really cater some of these assays for some of the difficult challenges that you face in the drug discovery, in particular the drug these interaction studies, things like specificity, species differences in response, trying to look at individual differences in the responsiveness as well. Okay. So just briefly, I want to describe a little bit about the nuclear receptors, and then hopefully this will make some of the subsequent slides easier to understand. So nuclear receptors are a family of transcription factors. In particular, these are what are called ligand-activated transcription factors. So they not only do they bind to DNA, but they are regulated by small chemicals that are able to directly interact with these molecules. As we'll see, many of these receptors have important physiological functions. One of them, of course, is to regulate drug metabolism enzymes, but they also regulate other important functions as well. All of these members of the nuclear receptor family fit into the family because of shared structure. And the main feature of the nuclear receptors is a DNA binding domain, which is called a C4 zinc finger. And I'll show you a little bit more about the structure and its binding in a second. But it turns out that in humans, there are 48 members of this family. And they all share a lot of similarity with each other. They have the same basic structure, but they differ in particular in regions that are important for their function. And they tend to classify or group in not only sequence similarity, but what their biological niche is. And so we find groups of receptors that are steroid receptors that are involved in hormone regulation. And then we have a big group of these nuclear receptors that are very much involved in regulating metabolism. And here's a little bit more detail about the structure and the structure to function relationships of these nuclear receptors. As we look from the amino to the carboxy terminus, we tend to describe the structure of the nuclear receptors as domains. And these are discrete units that have discrete structural features to them. The two main features of nuclear receptors are the conserved DNA binding domain, which is also called the C domain, and then a ligand binding domain, which is a series of alpha helices that forms the ligand binding pocket. So each receptor within this family has a distinct DNA binding domain that's able to interact with specific types of DNA elements, which are often called response elements. They have a distinct ligand binding domain, so they can recognize different small molecules. And I'll describe a little bit more about that binding in a second. And then there are other features within this molecule that tell it whether to homodimerize, what to heterodimerize with, and also other protein-protein interaction domains. Again, these receptors tend to fall into subfamilies. And so we have the steroid hormone receptor family, which tend to form homodimers, bind to their distinctive response elements, regulate target genes. We have a whole group of RXR heterodimer partners shown here with PPAR and RXR, and then some other orphan receptors that we know less about. We're going to be focusing more on the steroid and the RXR receptor families, and at the very end we'll talk about a non-nuclear receptor that's a member of this kind of broader class of ligand-activated transcription factor. So briefly, how do these receptors work? So we have these two domains working in concert, the DNA binding domain and the ligand binding domain, but there are many other factors that make these into workable transcription factors. So not only do they interact with DNA, but they interact with a whole series of different chaperone proteins and members of the nuclear receptor co-repressor or co-activator complex family. So generally speaking, in particular with these RXR heterodimer partners, these receptors in the unliganded state are bound to DNA, and they're inactive. They're not regulating their gene expression. They're not regulating gene expression, but they are bound to their response element. Steroid receptors are in an inactive complex in the cytosol, and they're being held there by a series of chaperone proteins. Now, everything changes when we have a ligand that specifically interacts with the ligand binding domain of these receptors. This binding causes a conformational change. In the case of the steroid receptor family, this causes a translocation to the nucleus where now it binds to the DNA. In the case of the RXR heterodimer partners, the ligand binding causes a conformational change, which causes it to lose affinity for the co-repressor complex and gain, gain affinity for the co-activator complex. So in both cases, we have this liganded receptor complex. We've now recruited this large group of proteins and enzymes to the site of DNA. And the end result is that we get the expression of genes that are specific for the ligand and for the receptor that ligand interacted with. Just want to briefly talk about binding is not the only effect that is important here. Your drug, not only does it bind, but it is causing a conformational shape change. And it's the shape of the liganded complex that determines whether you have an agonist, partial agonist, or an antagonist. So it's shown here in the left column, left side of the slide, is the estrogen receptor ligand binding pocket. In the case of an unliganded estrogen receptor, we have one of these helices called helix 12, which is buried in the inside of the receptor. So it is unable to recruit co-activators, and so we have an inactive complex. When a ligand interacts, such as estradiol, this causes a conformational shape change, which now the helix 12 is accessible to the co-activator complex. Now we get regulation of gene expression. If we have an antagonist like this ICI compound or something like tamoxifen, now this helix 12 is in a non-permissive location. You get no recruitment of the co-activator complex. And then we have the case of something like genistein, which is a phytoestrogen. And in this case, the helix 12 is in between what you see with estradiol and with the ICI complex. And what this does is it leads to a less favorable interaction with the coactivator complex. And so if you look at the different conformation, these result in different dose response curves. And this will be important when we talk about a pharmacodynamic interaction. But we can actually, with different chemicals that bind, we can have a full agonist, which leads to 100% activity of that particular receptor, a partial agonist, which even at high concentrations cannot reach the full maximal effect. A full antagonist in the absence of an agonist has no effect. You have no helix 12 available. And then an inverse agonist is kind of a new flavor of agonist that has been described. And in this case, what happens is a conformational change reverts back to the structure of the receptor that now actively recruits a co-repressor. Now, we're going to take advantage of knowing how nuclear receptors work to try to develop rapid screens to ask the question, does my chemical interact with one of these important nuclear receptors? And there are two major types, and I'm just briefly going to describe them. The first is to take advantage of knowing what the response element is that your nuclear receptor of interest or transcription factor interacts with. And you take that little piece of DNA and you put it upstream of an artificial gene. In this case, we're showing it as luciferase, something that's very easy to measure. We can take this plasmid, this purporter plasmid, transfect it into a cell line that expresses your nuclear receptor of interest, or we can co-transfect in that full-length receptor. And then when we add our chemical of interest, we are going to get not activation of our target gene, but we're going to get activation and expression of this purporter gene, something very easy to measure. It's utilizing all the same mechanisms that the endogenous gene promoter and enhancer elements are using, but we're just tricking it to regulate a gene that's easy to measure. There's another type of assay that's called a cis-trans assay that takes advantage of the modular structure of nuclear receptors. In this case, we can isolate each of the different domains and look at them individually. In this case, what we're doing is isolating the ligand binding domain, that EF domain, fusing it to a heterologous DNA binding domain, something like the yeast transcription factor GAL4. And now our reporter gene has the GAL4 response element driving luciferase. We transfect in our reporter gene and the plasmid containing this chimeric receptor. Now when we add ligands for the receptor, now we get very specific regulation of this particular construct based on its interaction with the ligand binding domain. And just a shameless plug here, Indigo Biosciences has a wide variety of both the classic and the cis-trans assays for nuclear receptors and other transcription factors. And we'll be using some of these assays as we move forward. Okay, so I want to just quickly talk about the pharmacodynamic drug-drug interactions, although I think this is fairly intuitive and easy to understand. So remember, this is a simple pharmacologic interaction between two different xenobiotics, both trying to occupy the same ligand binding domain of our nuclear receptor or ligand activated transcription factor. Now, in the case of nuclear receptors, this is important because nuclear receptors are important drug targets. So pharmaceutical industries have taken libraries and looked for activators for all of these different nuclear receptors to try to create drugs that are treating a variety of different diseases. And roughly 15 to 20% of all prescription drugs have as their primary on-target target a nuclear receptor. And this is just a list of kind of the top drugs that target nuclear receptors. There are many that are in clinical studies and preclinical studies right now. Another reason why this is maybe of interest is not only are these nuclear receptor drug targets, but they're also pollutant targets, nutritional targets, and microbial products. So again, there are several avenues of drug-drug interactions at the pharmacodynamic level that might be of interest. And here's one example of a pharmacodynamic drug-drug interaction that is targeting a specific nuclear receptor called Farnesoid X Receptor, or FXR. One caveat, and one of the reasons why I talked about agonist and partial agonists previously, is because this really determines what type of drug-drug interaction you're going to get at a pharmacodynamic level. So if you have two drugs that are both agonists for a particular target, then your perpetrator drug is going to be additive with the victim drug. They're both interacting with the receptor. The receptor doesn't care whether it's drug 1 or drug 2, it's still going to be maximally activated. If your perpetrator drug is a partial or weak agonist and your victim drug is a full agonist, then you will see a partial or weak antagonism when these two drugs are given together. And of course, if your drug is a full antagonist, then drug 2 is going to block the effects of drug 1 or your victim. Shown on the right over here is an example of FXR with a full agonist, this GW compound, a partial agonist, which is a bile acid CDCA, and then a full antagonist, this glucosterone, which is a natural plant product, where by itself it has no effect. CDCA cannot lead to a maximal response. GW leads to a full response. Now, if you were to activate FXR at about 80% of maximal level, and now do a dose response and increase the antagonist, what you'd see is a dose-dependent decrease in activity. So again, this is a classic pharmacodynamic drug-drug interaction. Okay, so for the rest of my talk, I'm going to try to focus on what is probably more complicated, and that is the pharmacokinetic drug-drug interaction. And this relies just on a real quick overview of what determines the pharmacokinetics of your drug of interest. And these are the basics of ADME. So how does your drug get in? For the purposes of this talk, we're just going to assume that these drugs are orally administered. Where does it go? How is it broken down? And how it's broken down is going to determine the concentration at its target site, half-life, potential bioactivation, or increasing and decreasing activity of your parent molecule. And then metabolism is also going to determine how it's excreted, which target organ it's excreted in. Now, the liver is seated at an optimal spot for being the site of metabolism, in particular for drugs that are given orally. And the reason for that is the small intestine through the portal vein goes directly to the liver. So the liver is the first site that sees the orally administered drugs. Also, the liver is highly vascular, and it's getting blood from the periphery through the peripheral artery. The hepatocyte, the main cell of the liver, is getting bathed in the contents coming from the portal vein from the small intestine. It's getting drugs or chemicals that are present in the bloodstream, the general circulation. They have a wide variety of drug metabolism capabilities. They're able to excrete it into the hepatic artery, ultimately to go to the liver for excretion, or they can pump it into the bile ducts for excretion into feces. So when we're talking about the main part of metabolism as it goes into these ADME processes, we're really talking about what's happening at the liver. And what the liver is trying to do to any xenobiotic is trying to take that molecule and detoxify it and excrete it. So what's shown here is a lipophilic xenobiotic, and the general process that a hepatocyte goes through in order to excrete it. And we'll go through some of these steps, but basically what it's going through is a two-step process to take something lipophilic, make it hydrophobic. These hydrophobic molecules then are substrates for these, for some pumps, transporters that are transporting into the basolateral membrane into the blood flow for urinary excretion, or into the biliary system to go into fecal excretion. All right. So the main part of pharmacokinetic drug-drug interactions occurs at the site of the hepatocyte. And what the hepatocyte, again, is trying to do is to detoxify this particular drug, going through a 2 to 3 step process, using a variety of different enzymes to first take this lipophilic molecule, make it polar, and this is done by enzymes such as cytochromes P450 and flavin monooxygenases. These polar substrates then become conjugated in the phase 2 system, and there are many enzymes that do this conjugation step: sulfonyl transferases, UDPGA transferases, glutathione transferases. And now these conjugates are much more water soluble, they're larger, and they're also good substrates for these transporters, which are part of the phase 3 system. And so these can be passive diffusion transporters like the solute carriers, or they can be active transporters like the ATP binding cassettes. So anything in our pharmacokinetic pathway where our perpetrator is affecting phase 1, phase 2, and phase 3 enzymes, this is the basis for a drug-drug interaction. And these perpetrators can be competitive substrates, so they're both trying to be metabolized by the same enzyme. They can be inhibitors, where the chemical is trying to block one of the key steps in this process, or they can be inducers that are increasing the expression of these enzyme families. So when we're talking about this pharmacokinetic, we're talking about a wide variety of enzyme families. Within each of these enzyme families, they have different substrates and different specificity that they're looking for. And this is just showing the phase 1 enzyme system, the CYPs. So these are all the different types of CYPs that are present in the liver. And these are all the different substrates that are being metabolized. So just quickly, if you know that your drug is being metabolized by CYP1A1, here already is a list of other drugs that might be potentially interacting. So we're already starting to kind of get a better idea of what chemicals can be victims and perpetrators of each. We can also we could generate the same table for each of these different families. They each have different structures that they're looking for. So how do the nuclear receptors fit into this paradigm? Well, really, where this is important is for that third type of perpetrator. This is the perpetrator that is regulating the expression level of these transport systems. And what I like to do is think of this as developing an adverse outcome pathway to try to understand this better. And for those of you who haven't been exposed to adverse outcome pathways, basically this is a predictive pathway where you are trying to understand the mechanism that a compound, a drug or a xenobiotic is leading to some adverse outcome. And you're trying to develop an understanding of linked pathways. So we have a series of key events that must occur before your xenobiotic leads to an adverse event. And the major steps are that first key interaction with some macromolecule. This is called a molecular initiating event, which leads to a series of linked key events. If these events occur, then your chemical is predicted to lead to that adverse outcome. So fortunately, we've been able to develop some of these pharmacokinetic drug-drug interaction pathways that have at its heart nuclear receptors. So xenobiotics, nuclear receptors, nuclear receptor ligands, activate a nuclear receptor, drive transcription. They're going to regulate a group of these drug metabolism enzymes at the mRNA level. This is going to increase activity of those particular enzymes, which leads to potential drug-drug interactions. Now, what's interesting, at least to me, if we go back to the left side here, is that quite often what we have is the xenobiotic interacts with a specific nuclear receptor as a ligand. Each of these different nuclear receptors is going to be regulating different groups of CYPs and phase 2 enzymes and transporters. Quite often, the ligands of the nuclear receptor drive the expression of the phase 1, phase 2, and phase 3 enzymes that are responsible for detoxifying the initial insulting xenobiotic. And so this is what's called the adaptive response, where these systems have kind of co-evolved with each other to recognize similar structures so that we can efficiently increase the expression of these drug metabolism enzymes to get rid of the xenobiotic that we have sensed. So these particular nuclear receptors are often called xenosensors. Okay, so that adverse outcome pathway now gives us kind of a good way of now attacking and trying to predict potential drug-drug interactions. So here is an advanced pharmacokinetic drug-drug interaction adverse outcome pathway, starting with the molecular initiating events. These key events are the enzymes that are induced, and then the adverse outcome is altering the kinetics of your drug of interest. A couple of things that I want to point out here is each of these different, each of the different molecular initiating events drive a specific subset of phase 1, phase 2, and phase 3 systems. So this goes back to that adaptive response idea. So I'm not going to go through all of the different molecular initiating events. I'm going to try to focus on some of the key receptors, molecular initiating events, and then try to talk about the nuances and some of the things that have made it more difficult. Let's start with probably the most important, and that is the pregnane X receptor, or PXR. Now, PXR has probably the largest ligand binding domain of all the nuclear receptors. So it's able to accommodate the widest variety of different structures. It's highly expressed in the liver, and so it really is kind of the classical xenosensor. It's in the liver, it's able to recognize a wide variety of different xenobiotics, which is shown over on the left here. All of these different drugs are PXR ligands. Now, after binding, these are all PXR agonists, and they're able to drive a subset of phase 1, phase 2, and phase 3 drug metabolism enzymes. All these different cytochrome P450s, UGTs, ABCs are all coordinately induced by the initial ligand. Now I have the CYP3A4 highlighted because CYP3A4 is probably the most important cytochrome P450 from a drug metabolism standpoint. It is responsible for metabolizing roughly 50% of all prescription drugs. So the substrates are, again, a wide variety of different substrates for the CYP3A4, but all of these other enzymes also have substrates that are being regulated as well. So in the classic sense here, what we're talking about now is our substrate, our drug, is the victim, and then any of these PXR ligands are going to be the perpetrator because we are inducing the CYP3A4. How can we use this information for drugs that we're trying to gain in understanding of drug-drug interactions? First thing that we can do is do a simple PXR screen and say, is my compound a PXR activator or not? And here's just a simple screening assay, a variety of different drugs, just looking to see, do we have full agonist, partial agonist of PXR? Again, trying to gain understanding. This is telling us that these have the potential for leading to drug-drug interactions. How else can we use this information? Well, we can use it to try to address some other aspects of evaluating drug safety, and that's trying to extrapolate between animal models and humans. And we know that drug metabolism is very species dependent. Well, so is the induction of drug metabolism enzymes. So what's shown here is a species different regulation with one PXR agonist called rifampicin, and its ability to regulate different non-human orthologs, such as monkey, dog, rat, and mouse. So simply what we did here is we took the ligand binding domain of each of these different species, put them upstream of or downstream of the GAL4 response element, and simply asked, as those small changes in amino acids going from human to monkey change the response of that receptor to the drug. And you can see here that with rifampicin, this is a good activator, a human PXR, monkey PXR, so not so active in that activator rifampicin, and doesn't touch the rat and the mouse. So your rat and your mouse studies are not going to see the induction of CYP3A4 that you would see in humans. So again, it's gaining understanding of whether you have to worry about cross-species extrapolation. And this type of extrapolation is not only important for PXR, but you see it with other nuclear receptors as well. Constitutive androstane receptor is in the same subfamily as PXR. This too is a xenosensor. It has a wide variety of different chemicals that it interacts with. This is probably the most difficult nuclear receptor to understand because it has different types of agonists and antagonists. It has a classic direct agonist. It has inverse agonists like androstane, and then it has a third pathway that I won't spend any time talking about, that is called indirect activators, where they're not ligands for the receptor, but they regulate the activity of CAR. And again, each receptor, different ligands, and regulates different genes. In this case with CAR, for the direct agonists, you have the ability to increase the expression of CYP2B6. Any substrate of CYP2B6 is going to be affected. Now, I did want to add a nuance here that in addition to all of those different types of ligands that we have for CAR, we also have splice variants for CAR. So these are just normally arising difference in protein structures just based on how the receptor goes from the pre-RNA stage to the RNA stage before it's translated. And in humans, there are 3 major splice variants that are seen. The first is the CAR1 variant, and this is a canonical isoform. It's constitutively active, and this is the form that you see inverse agonists with. CAR2 is just a 3 amino acid insert in the ligand binding domain, and this completely changes the activity of the receptor. It is no longer constitutively active, and now it's activated by xenobiotic ligands such as DEHP. DEHP does not touch the other 2 variants. So just the 3 amino acid change is enough to change its ligand binding specificity, but not its DNA binding specificity. And then finally, CAR3 is the major isoform for the direct agonist, and this will activate the prototypical activators such as CITCO in human and TC-BOBOP in rodents. So interesting that you have these different variations within an individual, and very simple to do to examine these different variants using the GAL4 system. We simply put in the cDNA for each of the variants into our chimeric system. We're finding that splice variants are seen in other nuclear receptors as well. Again, this could determine ligand specificity. Okay. The last example that I'm going to talk about is not a nuclear receptor. This is called the Aryl hydrocarbon receptor, and this is a member of a different family, but it really acts like a nuclear receptor. So it is a member of the basic helix loop helix POS family member of proteins. Is present as a chaperone complex. When it gets activated by a ligand, it translocates to the nucleus, binds to its response element as a heterodimer with another protein called ARNT, recruits coactivators, and regulates drug metabolism enzymes. In this case, what it regulates is cytochromes P450, the 1A1 and 1B1. And again, in a classic sense, this is a typical AHR ligands, which can be endogenous agonists, endogenous inhibitors, or drugs, are then able to regulate the metabolism of CYP1A1 substrates and affect their ADME. The nuance that I want to talk about here is something that we are just starting to gain an appreciation for. And that's how the microbiota are very important, not only for drug metabolism, but also for regulating many aspects of the drug discovery process. So, of course, the gut, the microbiome is very complex. Many different organisms are there. The mixture of the microbiome is determined by diet, genetics, pre-exposure to pharmaceuticals, antibiotics, other factors, obesity, affect the composition of microbiota. Why does this matter? Well, it turns out that nuclear receptors such as AHR are sensing different microbiota products. In the case of Aryl hydrocarbon receptor, it's responding to tryptophan metabolites. And they're responding to these different type of metabolites. And this AHR induction is trying to control gut motility and then trying to control and try to maintain a healthy microbiota. So one aspect of this drug-drug interaction is by a dietary factor or pharmaceutical changing the microbiota, able to induce AHR activation through these tryptophan metabolites, driving CYP1A1 expression, which again is going to be a drug-drug interaction. There's another type of drug-drug interaction that we're just beginning to understand, and that would be having a drug that interacts with AHR, which is going to affect the microbiota, which that by itself may affect drug metabolism. And it turns out other nuclear receptors are also involved in this maintaining a healthy microbiome, responding to microbiotic products, receptors like FXR, PPAR, PXR, and VDR. Okay. All right, so let's wrap up in our last couple seconds that we have here. So we talked about nuclear receptors as being key for looking at drug interactions, drug-drug, drug-nutrient, environment, and microbiome interactions. When we think about this, we want to gain as much understanding as we can, try to use these adverse outcome pathway maps to learn more about perpetrators as well as victims. We know that nuclear receptors are involved in the pharmacodynamic drug-drug interactions because they're primary drug targets. We also know they're very important for the pharmacokinetic drug-drug interactions because of these xenobiotic sensing receptors that are present in the liver, things like PXR and CAR. And how these fit into the pharmacokinetic pathway is through these receptors specifically regulating subgroups of the phase 1, phase 2, and phase 3 metabolism enzymes. One last thing that I'd like to end with is these in vitro tools that we've discussed are really helping you with prioritizing, screening, labeling, biomarker discovery. But of course, this is just predicted. We still have to go through and actually try to better understand whether we are changing the metabolism of our victim upon treating with the perpetrator. All right. So with that, I'd like to thank you for your attention, and I'd be happy to answer any questions that you may have in the last seconds that remain.
Okay, well, first I'd like to say thank you very much, Jack, for that insightful presentation. We'll put on our webcams. And just like Jack said, we have a few minutes left that we can try to fill in some questions from the audience. So audience members, if you like, you can still send in your questions using that questions window, and we'll try to get to your questions, or the team will follow up with you after. So let's grab one question I think we have time for. Here's the first question here. What is the difference between an antagonist and an inverse agonist?
Yeah, that's a question that we actually get quite a bit as we're trying to help people design studies. So an antagonist is a pharmacological interaction between an agonist and another chemical at the ligand binding pocket. So the antagonist is actually precluding the agonist from binding, which makes it unable to recruit a coactivator. An inverse agonist doesn't require the co-administration of an agonist. What it's doing is binding to the ligand binding pocket and then actively recruiting the co-repressor complex. And it does this in a dose-dependent way. And the dose response curve looks like the inverse of an agonist curve. That's why the name is an inverse agonist.
Okay, I'm going to squeeze in one more question. This audience member is asking, do you integrate pharmacogenomic in the assessing of PK/PD?
The pharmacogenomics can be incorporated, although we haven't done that. The approach that we used for the SNP, I'm sorry, for the splice variant, is something that can be used for looking at different polymorphisms. And so it'd just be a matter of incorporating in the different variants into the ligand binding domain, for example, and seeing how that affects the sensitivity to ligands. And we have done that just preliminarily with the PPAR alpha, one of the major variants within the PPAR alpha ligand binding domain.
Okay, all right. Well, that looks like that's all the time we have for these questions. So I'd like to say thank you very much to our audience members for sending in those questions. We have reached the end of the Q&A portion of the webinar. If we couldn't attend to your questions, and there's quite a number of them, the team at Indigo Biosciences, Inc. will follow up with you after this presentation. And if you have any further questions or inquiries, whoops, that went a little too fast there. Here you go. jackvh@indigobiosciences.com is the email that you can use, and Jack would be very happy to answer your questions. So thank you everyone for participating in today's webinar. You will be receiving a follow-up email from X-Talks with access to the recorded archive for this event. A survey window will be popping up on your screen. Your participation is strongly encouraged and very much appreciated, as it will help us to further improve our webinars. Now, in a few seconds, I'm going to send you a link in your chat box. You'll be able to view the recording of this event at that link and also share this link with your colleagues once they register for the recording as well. So I encourage you to do that and get even more out of today's presentation by downloading the supporting material available under the Handouts tab. And there's one there for you. Now, please join us in thanking our speaker, Jack, for that very insightful presentation and for answering your questions. So thank you, Jack.
Good, thank you.
Okay, we hope you found this webinar informative. It has been my pleasure to be your webinar moderator. On behalf of the team here at X Talks, we thank you for joining us. I'm Sonia Hunt. Until next time, please take care and bye for now. Thank you, everyone.